Dyadic is a very interesting company. We are not the typical biotech company that you would normally see. We have a platform technology, so it's multiple shots on goal. We've developed a technology platform over the last 2.5 decades related to the industrial biotech sector. You'll hear about that. We made enzymes at massive scale, very low cost, and we sold that to DuPont for $75 million and kept the cell line to apply it to human health and animal health to lower the cost, speed of development, and bring affordable health care to people. As you can see from our balance sheet, we had $37.2 million in cash and investment-grade securities as of 9/30, the last Q. We have no debt. We have no warrants, so we don't have any overhang. The other nice part about this is me and my family own about 1/3 of the company, so we're actually paying attention to the cash because a lot of that cash is our own, which is again atypical because biotechs burn money. We actually try to accrete our shareholders and are very sensitive to dilution. So we do things slowly, methodically, thoughtfully, and we don't throw darts against the wall. We actually have purpose for the cash and we apply it, which we think, in a systematic manner. It's interesting because we're here in London, and of course, I'm an American, and the office is based in Jupiter, Florida, right near Scripps, because Scripps is also in Florida, in case you guys don't know that. But we're really European in most ways because the industrial biotech sector really started in Europe with companies like DSM, Danisco, Novozymes, et cetera, BSF. And we have research facilities and collaborations in Helsinki, Finland, in Madrid, or outside Madrid, Spain, and our chief scientific officer is in Budapest. And the reasons that we're located here is filamentous fungi, which is the cell that we use, that we spent the last 2.5 decades creating, engineering to make massive amounts of low-cost protein at incredible scales, up to 500,000 liters. Is a European phenomenon. It wasn't an American phenomenon. It's now becoming a worldwide phenomenon. And we're sort of leading that charge in the animal and human health space. In the industrial space, fungi are the workhorses of the Novozymes and the DuPonts and the Daniscos and the DSMs and the BSFs. And several of them are using this fungi specifically. As I mentioned, we licensed it in the past for biofuels to Avangoa Bioenergy, which is a multi-billion-dollar company. BSF, well, I'm sure you all know, was also a licensee of ours. Small company, Shell Oil, spent $400 million on this technology, so we avail over that power, and that's why we're in Budapest. Our chief scientific officer ran that program, or part of that program with Codexis when the Shell investment on that. In Helsinki, Finland is VTT, it's the Finnish Institute of Research, and they are engineers of microbiology of cell lines for over 3.5 decades. And they avail over the engineering of the cell line. So we're really European, we're with a little touch of America. So here you can see we're on the NASDAQ. We have had a very nice uplift this year, and of course share appreciation along with that. We're gaining more and more attention both in the industrial world in terms of pharmaceutical animal health companies, with investors, with partners. We have 7 collaborations, if you count animal and human health, with top 25 pharma companies. People like Sanofi, top 5. 4 other unnamed top 25 pharma companies on the human side. 2 top 5 animal health, biologics, companion farm animals. Just signed a deal about 10 days ago with the second one of those, and that's advancing that program very nicely. Our members of the board and management team will get into shortly, and I think everything else we've probably gone over already. So I'm, of course, the founder of Dyadic. It's an interesting history. We don't have enough time to get into it, but I can tell you I started the Stonewash Blue Gene business selling pumice stone, transitioned to being a distributor of Novozymes and Genicor of enzymes when that came, developed our own enzyme technology, hired a bunch of scientists. They obviously spent a lot of money and time to do it. And 25 years later, here we are creating massive amounts of proteins at very low cost. And so Matt Jones, who's in the back of the room here, is based in London. He's our business development head, chief commercial officer, worked with Lonza, GSK, Ping Rawson, our CFO, and Dr. Cella, we talked about. He worked at Teva and with Codexis and Shell on that program. So he's engineering and scaling C1 as well. Our board of directors, it's very interesting. Most biotechs have a scientific advisory board and a board. Because it's my money, I'd rather spend it on one place. So I combined them. Kind of a unique approach. Seems to make sense, but apparently not in the biotech world. Anyways, we have business development guys that have science backgrounds from Merck. Dr. Buckland spent 29 years at Merck, chairman of the board of the National Institute of Manufacturing Biologics, U.S. National Academy of Science engineer member. Dr. Bose, equally skilled, worked at Pfizer 34 years, again, U.S. National Academy of Science engineer. And Mike Tarnock is the chairman of the board, comes from Pfizer. So we have big pharma, we have biomanufacturing experts. These are people that are fine-tuned for exactly what it is we need to be done, is engineering the cell, tweaking a proven industrial workhorse to move it into the pharmaceutical area so that we can bring affordable health care to people. Corporate achievements in 2019, in the last 18 months or 19, 20 months, I think we've done 15 deals, collaborations, some with big pharma, some in animal health, big pharma, some small biotechs. We work with the Serum Institute in India. We'll get into that later. It's one of the largest vaccine companies by volume on the planet. I don't know if you're familiar with them or not, but if not, I suggest you look it up on the internet because they're the real deal. And then we also own small equity pieces in small biotech companies that couldn't afford an upfront license and access fees. And in this case, we're sort of like OrbiMed or Deerfield or Wellington on steroids because they'll put equity in with cash. We put technology in without cash. We get equity. We speed the development and lower the cost, potentially, of their products, which increases our equity. And we get a kicker, which they don't get. We get milestones and royalties. So it's a great opportunity to expand the use of our technology with little biotechs that can't afford to get in the game up front. Rather than ignore them, we empower them. So the markets are obviously quite big. I think you're familiar with many of these things. Multi-billions, I think there's $320 billion worth of biologics being sold today. They're too expensive, they're unaffordable. 2% of the American population has access to biologics. It's 40% of the budget. I don't know anywhere in the world anybody would put 40% of their money to get a 2% return. That's got to change. And we think that until you change cell lines, that's not going to really make a big dent in the problem we face as human beings. So it's really all about expressing proteins, taking a piece of DNA, putting it in a cell, and making a protein. And that's what we're really good at. This is where we spent years in the industrial biotech sector. We work with a fungus called filamentous fungi called Mycephalothora thermophila. We nicknamed it C1 for short. It's a lot easier to say. And C1 is very unique in a lot of ways, and we're going to get into that. So it's low cost, high yield, quicker, faster, cheaper, more affordable, low cost media. We've already sequenced the genome many, many times. We know its genetics. We know the metabolic models. We've sort of metabolic pathways. You'll hear something about that later on. And we think that we can dramatically disrupt the way biologics are developed and produced. So we sold our industrial division, which we talked about, for $75 million to DuPont 3 years ago. We had a theory. Our theory was we thought we could make antibodies, vaccines, virus-like particles, antibody fragments, Fc fusions. And our theory turned out that actually we did. And we made them at very high levels. But it's really all about the cell line. Because in the end, you have to take a piece of DNA, a gene, put it into a cell to make a biologic. So let me tell you why this cell line is unique and novel. And it's because filamentous fungi are nature's most productive secreting organisms. That's what they do in life. We're not forcing something to do something it doesn't do naturally. We're just ramping it up and producing more and pushing it harder. And we did that for 2.5 decades. And as I mentioned, there's been about $500 million spent between Shell Oil, Codexis, Dyadic, DuPont, BASF, Abengoa Bioenergy on this technology. And what happened here is we had a serendipitous mutation early on that just random mutagenetic before the sequencing of the human genome. It changed the structure, the physical form of how this fungus grows. That turned a long hair into whiskers. We had 300 tips instead of 2. We improved several hundredfold the productivity of this just on that one mutation. It was done randomly, so we don't even know how it was done today. but we can replicate it, so it doesn't matter. We've been replicating it for more than 2 decades, so it's not like we're going to lose it. And then we spent all this time to develop the molecular tools, how to program, put DNA in, take DNA out, and to modify the cell to make the things we want. We can virtually make any biologic you might want us to make or our partners want to make by engineering these cells, but they're hyperproductive cells. The background of the cells, we've removed a lot of the background proteins, so it's cleaner when you produce what you want. It grows faster, it grows quicker, and it grows on glucose and amino acids, so it's cheap. The media is like 1/20 of the cost of a CHO cell. So it grows in half the time with 1/20 of the media at 2 to 5 times the yield. So when you combine all that together, it's just revolutionary. It's potentially disruptive. And the pH and the temperature ranges are much wider, so it's more robust, it's more versatile, it's easier to work with, it's scalable. We're actually scaling down. We grew at 500,000 liter scale. The largest Samsung plant's 15,000 liters, 1/35th the size of what we've been growing at. And because it's more productive and it grows quicker, we're actually shrinking it from that down to maybe 2,000 liter fermenters and getting the same output that you would get out of a 15,000 liter fermenter with lower cost. So one of the things we didn't know is, could we grow this filamentous fungal cell in a single-use bioreactor? We grew it in stainless steel tanks for 2.5 decades. We knew that. We knew we could do robust, versatile, efficient. The answer is yes. We tried it. We leased a XBR50 single-use reactor from GE, and we ran 6 runs and demonstrated equal productivity in these plastic bags with an agitator. So it gives you more flexibility as well. You can grow in stainless steel or in single-use reactors. So again, we applied this as we talked about the theory. The theory was, could we take this productive host that was developed for an industrial world and bring it to an inefficient pharma world? And the answer is yes, we did. And we hit record levels of productivity. We've hit 22 grams per liter in 168 hours. That's compared to that's what, 7 days versus 14 days. So CHO cells would be 14 days. The average production is 4. So if you take 22 and you double it, it's 44. That's a huge difference. So from a productivity standpoint, these are really, really important figures. The antigen in the middle down below is the last one because of time we can talk about. There's an EU program called ZAPI, Z-A-P-I. If you look up ZAPI on the internet and Dyadic, you'll see who's involved in that. There's 20 different organizations, commercial partners, academics. The target was 100 milligrams. We hit 1,700 plus. The nearest competitor was baculovirus insect cells. They were at 50. So you can see they were 35 times more productive than that. So that's why the animal health people are all over this right now. They see the productivity of these vaccines, the virus-like particles, where cost of goods is dramatically important in an animal health, whether it's companion animals or farm animals. The binding, the quality. The binding is virtually identical, whether you produce it in C1 or you produce it in CHO. This is an interesting, bispecifics or these multi-specific genes are getting difficult. Some of these things are very tough to produce. So if you can't produce enough, you can't even do research, let alone commercialize it. Some programs get killed because of that. We can bring them back to life or keep them alive, and then ultimately you can make them more affordable to people because you're making it for a lot less. In this case, the cancer cells were killed virtually identical from CHO cells produced bispecific or C1. So again, as I mentioned earlier, we're working on our own sort of biosimilars. You'll see that coming up. A certolizumab antibody fragment, which is pegylated, which is like UCB here in England. They're 1.4 billion euro drug, Cimzia. And then we're working with Divo, which is expected to be about an $8 billion drug in the next year or two from BMS. And then we have small equity positions, as we mentioned earlier, in these little biotechs who can't afford to take an upfront cash payment to get a license. But instead of ignoring them, we're empowering them and enabling them to speed the development, lower the cost. We get equity and we get milestones and royalties as kickers. So versus a Deerfield or OrbiMed or Wellington, you might think about Dyadic having a much better return on your investment just on that side of the business alone. And we'll grow that to more and more of these little biotechs. So one of the things we talked about is the Serum Institute. I'm going to go through this quickly because I'm running out of time. This is a company that's been around 50 years. They're the largest producer of vaccines in the world by volume. They have a program with us for 8 antibodies and 4 vaccines. And this is somebody that's actually pushing us forward to try to maybe even expand the 12 proteins and do other ones. Certolizumab, we talked about as a biosimilar to Cimzia. We're working on that. We'll get into that in questions. We're working on the downstream processing. Divo, we briefly talked about. Same thing here. Working on that as a biosimilar to demonstrate that we can make glycoproteins in C1. We're glycoengineering this. In fact, tomorrow in Lisbon, there's a PEGS conference, Protein Engineering, and there one of our scientists from VTT is presenting on our glycoengineering, and I think that's exciting results, so you can stay tuned for that. And then we've developed a way to make the seasonal flu vaccine quicker, better, and cheaper. Through mice trials, we can use 1/3 the amount, get a better result, and produce it for less. And then one of the last things we're doing is we're trying to use this technology to see if we can make AAV viral vectors, because if we can do that, we can dramatically open up the bottleneck, because as you know, there's unaffordable, their productivity levels are low, there's not access, so that's another great opportunity. And then we're working on, last but not least, on primary and secondary metabolites, which is pathway engineering, synthetic biology, and this cell line, again, is a proven industrial workhorse. So if we can engineer the pathways in that are necessary, we can be the low-cost producer of some of these products. And I'd say stay tuned as we announce what some of those are we're working on. And I think just as a recap, again, we're well-funded, no debt, no warrants, no overhang, great management, phenomenal technology, and we're just applying it in a whole new world in a lot of different ways, and it's a platform. So thank you very much.